Hello student, you are welcome in Enlightenment Agri Academy. So today we'll be discuss on the mint diseases. Mint is a medicinal crop. So now we'll discuss on the various diseases caused on the milk. In this lecture we will discuss the crop disease and their management in mint. So basically we study here the mint anatomy. The it's it's botanical name of mint is Mentha species. So now here we will discuss the above part that is called as the inflorescence. The floral part of mint is called as inflorescence. So now here inflorescence has a uh, white to purple flower in cluster, that is the verticillium on erect spikes, two leafed corolla, small dry and dry capsule fruits is set on this inflorescence. So now second one is the leaf part, and that leaf part is called as foliage part. So it is a opposite pair, that means leaves are arranged in a opposite pair, such like this one leaves and this one is, they are the both in opposite direction. So now the simple oblong to the lanceolate downy structure, serrate margin, dark green, grey green to purple blue colors are in this foliage or maybe leaves. Then third one is the root system. It is the wide spreading underground rhizome, or it is also called as stolons. So now it is spread by stolons. Then its multiplication by stolons. Then its one is the stem. Stem is the erect, distinctively square and branch. That means this is the branch. This is the main stem, and they have lots of branches, and they are multiplied. Or maybe it is the primary economic value expected for menthol, production of menthol. Then menthone. Third one is methyl acetate, terpenes, almost and this plant is the perennial in nature. That means its life cycle is more than 2 to 3 years. So that plant is called as perennial plant. So now the various diseases was caused in mint. So such one as the powdery mildew, iricipi, chicoracerum, then rust is the Puccinia menthi, leaf spot Coloraria lunta, and then one is the leaf blight Alternaria alternata. That disease is caused, and then also is the wilt, that is the vascular attack, that the verticillium wilt is causing this. In the root part, that is the rhizome and soil attacker. One is the stolen rot, and that rot is caused due to Rhizoctonia bataticola and uh, Sclerotium rolfsi. Then we will study here the life cycle of these systems. In the sexual stages, first one is the optimum temperature, high humidity is the trigger the sexual asexual stages of this uh, pathogen. Then primary inoculation or dormant mycelium is present in the soil, or maybe on the debris of the plants. Then sclerotial bodies, they germinate and cause the diseases on the first primary on the first plant. Then in the spread, this first plant produces so many conidia, and that conidia is transferred to the second plant, and that is called as the secondary infection or secondary inoculum. In the massive asexual reproduction via the uh, ear earborne airborne conidia, then or soil-borne mycelium, mycelial strain to drive the rapid secondary infection. Uh, then uh, adverse climate condition. If the climate condition is not favorable to the development of the disease, then the high or low temperature or unfavorable uh, relative humidity, then from this mechanism, asexual mechanism is convert or shift into the sexual reproduction. That is the gametophysical contact between the two gametes, then to formation of plasmogamy, then karyogamy, and then comes the meiosis and uh, production of the spores. Outcome: creation to highly resistant sexual spore, sexual to survive until the condition improve. Then we will study here the disease, that is the powdery mildew. The pathogen is the Erysipecis serum. Then its taxonomy is the kingdom fungi, division Ascomycota, class Ascomycetes, uh, and Erysipe. That is the family. Genus is the then favorable for epidemiological trigger, that is the 28 to 32 degrees Celsius is favorable temperature. Then 80 to 86% relative humidity, warm and cloudy weather will be favorable for the disease development. Then etiology: external septate mycelium or subepidermal hosteria, they can cause the diseases. Then spore, they produce the asexual spore, that is the barial conidia or on oidium, Panarcoidea on oidium, and then sexual, that is ascospore in the Clistothecia. Clistothecia is the completely closed fruiting body in the powdery mildew. They have the three types of the fruiting body in the fungi, that is the Clistothecia, then second one is the Perithecia and Apothecia. Apothecia. These are the three fruiting bodies were present in fungi. So here only the Clistothecia body, that is the completely closed body is present. Then inoculum, that is the primary dormant mycelium and secondary from the airborne conidia. If the downward mycelium is present in the soil, then they cause the primary infection to the first plant, and then after the plant produce the number of conidia, and that conidia transport to one plant to another plant by the winds. So here we study here the visual symptoms. Firstly, the small chlorotic spots on the upper lamina, then upper surface of the leaf, then form the small chlorotic spot. Then second one is the brownish discoloration on the lower surface, and then heavily white gray powdery growth leading to the total defoliation. And that is the three basic symptoms produced by this powdery mildew fungus. So here the what is the management intervention was need to control the diseases, that is the culture, that is the crop rotation with the other crops, then alter the sowing dates, ensure the optimum spacing and nutrient to improve the air flow. Then chemical treatment, that is the prophylactic aerial spray, that is the wettable sulfur, how much, that is the 0.3% was used. Then carbendazim, that is the 0.1%, and or third one is the calixin 0.15% at the interval of the 10 to 15 days. That is the two spray was recommended for this powdery mildew control. Then second disease, that is the major disease in the mint, that is the rust, and that rust is caused due to the Puxina menthe, is the causal organism of this rust. That the taxonomy, it is also the kingdom fungi, division the Basidiomycota, then class is the Basidiomycetes, and Puxinaceae is the family, and genus is the Puxinia, and species is Menthe. That the favorable temperature, that is near to the cool temperature, that is the 70 to 27 degrees Celsius. Relative humidity is 90 to 92 percent. Long days hour, that means it is a long day hours, and 1 to 2 hours morning dew. That things or that condition was favorable for disease development of rust. Then etiology, it is the septate intercellular mycelia or intracellular bacteria. They can cause the etiological space. Then the spore, they also produce the sexual spore. That sexual spore is called as eudospores in uridium, and sexual pore is the teliospore in the telium. Telium is the body to produce the teliospores. That is inoculum. There is the no inoculums was seen. So here study the visual symptoms. The circular brown rusty pustules on the lower surface of the leaves. See here, that is the brown or rusty pustules on the uh, lower surface of the leaf. Then Telesia stages swelling on upper stem portion. It's just like swelling of this portion. Then outcome, see where the leaf blighting and that chlorotic sticking on upper surface. Just like the chlorotic sticking was seen on, and then outcome severe leaf blighting and that of the leaves. So here the management strategy: strict crop rotation with the non-host resistance and foliar spray mancozeb at the 0.25%, and then hexaconazole 0.1% is effective for the control of the diseases. Then third one is the leaf spot. Leaf spot is also major diseases in the mint. That the clinical base, the Coluaria luenta is the pathogen. That the favorable condition is the 28 to 30 degrees Celsius. Relative humidity is 85 to 90%, and cloudy weather, which that is the cloudy weather is favorable for the disease development. Nutritionally poor soil and neglected crops are the variable key for this disease development. Then etiology: septate mycelium, intracellular or intercellular hostilia. Color conidia is the specific characterization of this leaf spot pathogen. So now here the spore that is the asexual spore, that is ear-borne conidia. Ear-borne conidia. And sexual, that the ascospore in the pseudothecium. Pseudothecium. And then primary inoculum is the dormant mycelium, and secondary one is the airborne conidia. About the rust, rust is I don't know the which inoculum was useful for the transmission of diseases. So now you study the visual symptoms. A small unclear brown spot scattered on the lamia. On this lamia, the small unclear spots on the lamia. Then collage into a massive spherical irregular space. That is small space, small spots is converted into the big spots, irregular patches, and the primary tip to no nutritionally star dense crop. So management strategy: the cultural destroy the affected parts, radically increasing nutritional status and reduce the plant density. Reduce the plant density. It one of the best method to control the diseases. Then biological application of trichoderma species, chemical spray, area spray, the mancozeb, that is the 0.25% spray was needful for the disease control. So now fourth one is the leaf blight. It is caused due to the Alternaria alternata. So firstly we see here the visual symptoms. That is the characteristics round, that is mean goal, then round, then oval, dark brown spot. Defining signal, that is the distinct concentric ring, and that concentric is the characteristic symptoms of this Alternaria fungus in every crop. We can see here also the mustard or may other crops. So see that is the concentric ring, just like this one. And this one is the basic characteristics of this pathogen. Then infection originated from the lowermost foliage. Lowermost foliage they originate the infection, and then outcome: spot expansion causes the complete brightening and leaf detachment. That is the outcome of the diseases. Then we study here the etiology: septate mycelium, color muricates conidia. Color muricate conidia, in which that is the color conidia is present also in the leaf spot diseases. leaf spot, coloraria, and second one is the leaf blight was caused due to the alternaria alternata. Then we can study here about the leaf spot diseases. Its need to color cornea was produced by both the leaf spot and leaf blight of pathogen. Then in a management strategy, for the management strategy, we can implement the standard prophylactic culture practices, ensure the proper canopy airflow, and disturb upward migration of the pathogen. Upward migration of pathogen. That means the first infection of this pathogen was caused in the lower part of the plants, and then it's transferred to the upward direction of the plants. So here, fifth one is one of the major diseases, that is the wilt disease. And that wilt is caused due to the verticillium alboatrum. It's the complete loss due to the these diseases. So epidemiology, that is the 30 to 32 degrees Celsius, and relative humidity is 90 to 90 2 degree 2%. Low moisture content is needed here. Soil trigger, alkaline pH, and black clay soil are the favorable soil condition for the this wilt disease causation. Then etiology, that the septate mycena, V-shaped conidia spore bearing round conidia, which round conidia, they are the produce. Spores or inoculum, ascospore as sexual spore, then micro and macroconidia, that the asexual and sclerotial body as produced by this this pathogen. That is the visual symptoms. First one is the unlaterate dropping and serve as the etiolation. Then V-shaped, see such a here, you can see here such a V-shaped, V-shaped yellowing on the lower leaves, V-shaped yellowing on the lower leaves, and internal, that is the light tan discoloration, just like this one in the vascular bundles. So here the management strategy: culture, burn the affected parts, utilize the nematode-free soil, and restrict the crop rotation. Crop rotation is also needful, and then also the nematode free soil. Then biological trichoderma species was used. Chemical, that is the drenching, deep soil drenching with 0.1% carbendazole is the beneficial for this control of the diseases. Then C is the sixth number, that is the stolen rot. It is also major diseases. Symptom was seen by that is the above stunted growth, yellowing, and complete wilt was seen in. Then underground, that the lesion progress from the pinkish brown, which that is the pinkish brown. Pinkish brown progress from to dark necrotic brown. Pinkish brown to dark necrotic brown. See here, you can see just like this one here, just like the pinkish color. Then terminal stage of the black patches or total soft decay of the stolons. Then management practices: total destruction of debris, deep summer plowing, and soil sterilization. These practices was used for the management of the disease. Preventive measures were taken by the farmers for the control of the disease. Then biological application of trichoderma for the chemical soil drenching with carbendazole 0.1%, or also the mancozeb you can use 0.25% per liter for the control of these diseases. Then synthetic arc or environmental trigger map, we can see here. So the danger zone for the basically four diseases or four pathogen, that is the about the 30, that means 28 to 32 degrees Celsius was favorable environment for these diseases. If they have the 28 to 30 degrees Celsius in environmental temperature, then that all the four diseases was caused. But in the cold condition, that is the about the 70 to 72% degrees Celsius temperature was effective to cause the rust diseases in the mint. So now the microclimate mandated 5 to 6 major fungal pathogens are nearly identical, that the 28 to 32 degrees Celsius and about the 85% relative humidity. Disease in the Mentha species is the fundamental as a failure of the microclimate and the canopy management. Avoid the dense planting, promote the airflow and aggressive disturb his localized humidity trap. Then we study here the comparative study between these all the diseases. Uh, the powdery mildew, they are the attack on the both the upper and lower leaves. Then that the white gray powdery patches were seen on the leaves. Then dormant mycelium, that the primary or clistotheria is the primary inoculum for the disease development. Then the rust, that is the lower leaves. Basically, rust is caused on the only on the lower leaves, or may also in the stem. Brown rusty pustules or stem swelling is the characteristics visual symptoms of the diseases. Primary loculation, they don't know. Then it's the leaf spot, that is the lamia. Uh, Target anatomy is the lamia. Spherical irregular brown patches was seen, and that is the primary loculum is the dormant mycelium. Blight, lower to upper leaves. That means they firstly they cause the diseases on the lower leaf and then go upward direction from lower to higher leaves. So that is the dark brown spot with the concentric. That is the characteristic symptoms of the dysarthronaria in every crop. Their primary location they don't know. In the wilt, vascular system or whole plant maybe die or death of this plant. There is a V-shaped yellowing just like on the leaves. That is the V-shaped structure or unilateral dropping was seen. That the dormant mycelium is the primary inoculation. In the stolen rot, that the rhizome rot or pinkish to black spot decay on the stolon, that the sclerotial bodies in the debris was presented, and that is acts as the primary inoculum for the disease development. So now here the integrated management. Firstly, there's the Ecuador response the chemical active. First, what is the foundation of the management? Prophylactic culture practices you can take on to control the diseases, and it's also low budget or minimum budget was required. That is the summer plowing. Then the strict crop rotation with the non-host, altering sowing dates. Then destroy the affected debris. Utilize the nematode-free soil. Then plant density is low. Relative humidity is also low. Then mitigates the dormant primary inoculum. Dormant primary inoculum. Inoculum. This is the you can take this preventive measure for the disease disease development. Then you can use the biological intervention, that is the trichoderma species application in the soil. Trichoderma species application in soil or root zone area. Then use as the primary leaf for the primarily on the leaf spot, stolen rot and wilt diseases they can control by this trichoderma. Then foliar, if the problem is acute or it may be with the problem was increases, then you need to control, that is the mancozeb, that is the 0.2%. Rate of mancozeb is 0.25%. For the rust and spot, it is the beneficial for rust and leaf spot. Mancozeb, we can say here shortcut mancozeb. Then wettable sulfur, that is the 0.3% for the control of mildew, that is the powdery mildew. Okay. Then hexaconazole 0.1% for the rust control. Then calixin, that is the 0.15% is used for the mildew, that is the powdery mildew is major diseases. In the soil drenching, that the carbendazim 0.1%, carbendazim 0.1% and mancozeb 0.25% is useful for specific vascular or stolen target. So now this way we can control or take the preventive measures for the control of the diseases. So now, student, your thank you for watching this our video for the lectures. So now, your support is the main. Like, share, and subscribe our YouTube channel for the more. We can take the lectures for the third semester student. This semester third semester student, that is the fundamental of hematology. And for the fifth semester student, that is the diseases, field and horticulture part and the management. So please share to the student. That will be the benefit for them. So now I have the one Telegram group or I have the our number. That is the useful for you if you have any queries regarding the this plant disease. Our number is 9796439361. You can message on this number and then we will be solve your problem.